Hi, everybody. My name is Negar Agah. Today, Dayanara Mendoza and I will be going over our project, which is evaluating tools for leaf nutrient analysis. And our mentor throughout this project was Bernardita Solar. Just a second. Yeah. Here we go. So a little bit about me. Like I said, my name is Negar, and I'm currently studying biological systems engineering at the University of California, Davis. However, I was raised in Iran. Currently, and I'm still here, but my internship took place at the processor station for WSU. So before we begin, I would like you all to imagine a scenario. Let's say you're trying to plant trees and you know that they're genetically identical. So you know that they have similar traits, but that doesn't mean your orchard is all going to be uniform and the same. Why is that? That's because there's inherent variability caused by phenotypical differences, right? So I don't know, your diameter size might be different, the shoot lengths can be different, all as a result of phenotypical differences. There's also another difference that I'm going to propose to you, which is just after planting, the microenvironment around each tree is going to cause certain changes. So let's say your water supply, your nutrient supply, they're all going to cause more differences. So there is a nutrient demand that's going to be different across your orchard. That leads us to our key question. How can we measure and manage this nutrient variability effectively? And why does this matter? It's essentially to be able to minimize the amount of input that we put into an orchard and reduce waste of input, because once we apply uniformly, well, certain parts of your orchard could use less. So what does nutrient management even look like today? What are most growers using to analyze and assess their trees? They're using what we call the standard procedure, and it's usually divided into two parts because you can't use a single test to confirm what you need to apply. That's why we use leaf tissue samples and soil testing. Both of these take place in a lab setting, so they're very much established and they can identify deficiencies for all essential nutrients. They're used in academic settings and commercial settings, and they usually take place once a year. So in our case, when this internship took place, it was around July to August. And the purpose of these tests is to represent entire blocks. And that's how farmers or growers can determine fertilizer rates using these tests that take place in the lab. But one thing you should keep in mind is that these tests tell you what you're deficient in, not a formula for what you have to apply. So that's what we're essentially trying to figure out in this project. So what are some problems with the leaf nutrient analysis, even though it's a well-established method? The first point is that it can be destructive and costly. So during our leaf collection, we had to be very careful with our samples. We also had to keep in mind that they do cause stress in leaves when you're taking away samples. The second problem comes down to narrow representation. We do get precise results. However, once you take samples from any orchard, and the standard is about 50 leaves per, say, 10 acres, you are going to get a composite sample from the lab, and that can reduce to some sort of representation that doesn't actually display every tree individually. And the third problem, which kind of explains the motivation behind this project, is just a demand for alternatives that we can try. We need results quicker, so we're looking for methods that might take less time and that can reduce shipping costs. So let's look at the big picture. The long-term goal for this project is to reduce orchard variability by implementing site-specific precision practices. This means that, yes, we're trying to treat each tree as though it were an individual one, and that is idealistic. So let's break this goal down into two more parts. The first part is exploring better indicators of nitrogen status, and I'll explain why we're looking at nitrogen later on. And we're going to use these indicators and use these tools to create prescription maps. What are prescription maps? Simply put, they're just digital field maps that can provide recommendations for growers for, let's say, what application rate they're supposed to apply. So now let's get into the methodology for our project. We analyzed 4 different tools, and the first one I'll get into in the slide after. Our methodology begins with the 2 different sites that we looked at for my portion of the project, which took place in Mattawa and Zilla. Both of these smart orchards have different cultivars of apples. Mattawa's was Cosmic Crisp and Zilla's was Envy. For each sample that we collected, we had to make sure that the leaves were mature. Why are we doing this? Just to ensure that we account for the standard amount of nutrients in a mature leaf, because here we're not testing leaf age. So typically young leaves can be very much variable in terms of the nutrients they have. So we looked at, as you can see in the image, leaves that are mainly mature, and we had to collect 20 per the 8 random sites that we chose, as you can see in the images. Now, So our main goal, the first goal, was to evaluate better indicators of nitrogen status. We're looking at nitrogen because the tools enable us to look at this nutrient in particular because it's a great indicator of application rates for growers. So we talked about the first method, the standard lab method that almost most growers use. The second one is also a well-established method, but the fun thing is that you can use it in the field. It's called the SPAD meter, and it's a kind of optical sensor. The two other new handheld spectrophotometers that we're going to get into were offered by Croptics and AgroCares. Unfortunately, AgroCares, we weren't able to test their scanner, and Croptics' product is still undergoing development. So first of all, why are we using a SPAD meter and how does this device work? What's the purpose of a SPAD meter? And there's actually literature sites establishing the reliability of this device. Before we get into it, we have to first understand how this works. As you can see in the image, the device works with an LED light and a detector. Once you put a leaf in between, the LED light shines two different wavelengths of light, which are NIR and IR. So that's near infrared and infrared light. IR tells you how much absorbing content there is. So once we put these two wavelengths and what the detector receives in the form of a ratio, you actually get what we call the chlorophyll concentration index. Simply put, that's just a ratio that is translated directly to chlorophyll. The good thing is that chlorophyll can actually be modeled to give us nitrogen. So this device gives you two values, not a full range of nutrients, which can be seen as a disadvantage. You're only getting nitrogen, and nitrogen is actually just received as a proxy by chlorophyll. Well, that's for the SPAD, which is pretty interesting. And Croptics and AgroCares, they also have in-field devices. And the interesting thing is that their devices are kind of right in between lab methods and the SPAD meter because they offer you multi-nutrient profiling. And they can also scan up to, at least for AgroCares, about 100 samples per day. So that's pretty efficient. However, both of these new technologies, much like anything else, were faced with a bit of skepticism in research because they require data validation and ground truthing. So as I mentioned, we didn't include these products in our research yet because we're relying on ground truthing at the moment. So how did our sampling occur? As you can see in the image, we collected 20 leaves. 20 of these leaves were ran through both commercial laboratory and also the SPAD meter. And the good thing about the SPAD meter was that we were able to connect it to a computer, and the computer directly input our data into Excel, so we were able to watch our show on the side. And of course, the standard laboratory method, we won't get into the details, but that is the process displayed above. And also we sent leaf samples to Croptic so that they can also validate their technologies for our ongoing research. Okay. So what results did we collect and what did we find out? This is the Mattawa site, and we actually found out that the ideal nitrogen percentage received by the lab was well within and above the ideal range. This ideal range is already established in literature research papers, and that's a good sign. That means that there's no nitrogen deficiency. And same with Zilla. So we found out that our 8 sites were pretty much sufficient in nitrogen. But we did find, interestingly, no correlations between the SPAD nitrogen and the standard method nitrogen. And this is something we'll get into later, but for now, we can assume that, okay, there's no correlation. It's also the same with Zilla. There's more explanations that we can get into this, and that'll happen later on down in our presentations. For now, I'm going to pass you guys off to Dayanara for her portion of the research.
Hello, everybody. My name's Dayanara Mendoza. A little bit about myself. A little bit about me. I grew up here in Washington State. I am currently going to Columbia Basin College. My mentor for the summer was Brenta Salato. One interesting fact about me is I've been working as a technician here at WSU for the past 2 years. So what is the green debate? SPAD versus standard nitrogen. Previous research has Previous research has shown that there was no correlation between standard nitrogen testing and SPAD testing. As you can see on the graph on the bottom right, we have no correlation and low R value, which leads us to the question, why is there no correlations? Our previous research from last year shows that we did find correlations. For example, on the left chart, we found a positive correlation of NDRE and leaf nutrients. On the right, we have a chart of SAVI and iron. In this chart, we have a negative correlation. This project took place at the WSU Rosa Research Farm in Prosser, Washington, where we worked on three cultivars: Wall 38 Cosmic Crisp, Honey Crisp, and Wall 64 Sunflower. I would also like to mention that these are all on G41 rootstocks. On the right side, you can see from a Google Images showing the blocks that we took samples from. From these cultivars, we selected leaves from 4 individual replicated trees, sorry. From each tree, 40 leaves were taken for analysis, and I also want to mention that these blocks have been managed by Bernita, where she applies very little fertilizer. Okay, sorry. So from each tree, 4 types of leaves were collected. We collect fully mature leaves from spur to seasonal shoots, with and without fruit. In this picture, you can see a spur with a fruit, vegetative with no fruit, and vegetative with fruit. Sorry. We only collect mature in order to have a standard. Once we collect these leaves, we used the SPAD meter that Negar has already explained, but to briefly explain, leaves were placed onto a sensor and the value was recorded from where we put it onto our computer and imported the data into Excel for data analysis. The same set of leaves were grouped and sent to a standard laboratory for leaf nitrogen testing. Results. This is a bar graph in the concentration of all leaf tissues by cultivar. The difference between tissues were not agronomically relevant. In this bar graph, I have placed a green box highlighted the recommendation leaf nitrogen levels that Negar mentioned earlier. As you can see, Honeycrisp and Wall 64 are within above and recommended values. Respectfully, while Wall 38 is below and recommended values. In the Wall 38 block, Bernita has been controlling vigor by reducing nitrogen applications, and we can see the difference by measuring the leaf tissues. Now on the left side of the slide, we have a linear correlation between SPAD nitrogen and leaf nitrogen concentration with an R-square value of 79%. As we can see, we see a wider range of nitrogen levels helped in better understanding the relationships between the two methods. To clarify, if we were to look at each cultivar individually, we will not find a strong correlation in Honeycrisp and Wall 64, which are the darker green dots. However, in the Wall 38 cultivar, we do see a linear response due to trees being deficient in nitrogen. In conclusion, the SPAD might still be a good indicator of nitrogen levels. However, that would not necessarily show up in a correlation analysis. When nitrogen levels are within accurate ranges, the values become irrelevant. If you are interested in knowing what environmental factors affect leaf nutrients, this is a great article to help explain them. And I'd like to say thank you to my mentor, Benita Salato, and the Salato Lab. And I'll be passing you guys back on to Negar.
All right. Thank you to Dayanara for explaining the reasons behind why we actually didn't see a correlation. So just to elaborate, the reason why we agronomically also can't see a correlation is because we're reaching a point that once you increase the amount of nitrogen, there is no visual symptom coming from the chlorophyll meter. So the chlorophyll meter essentially tells us the greenness of leaves. That wasn't indicated in our graphs, and that's why we didn't see a correlation, because no matter how much nitrogen you apply, you're not going to see a statistically significant reason. So moving forward, what's our next goal for this project? Like we mentioned, it was to develop nutrient prescription graphs. We can do this using aerial mapping tools and also different tools that we used in the field. There is a formula, and most growers use this, to find the rate of application of nitrogen in particular. But how are we going to find the rate? There's 2 main components, and the first one is demand and supply. How do we find demand? Keep in mind that in apples, new fruits and shoots account for about more than 90% of total nitrogen demand. So this means we can actually correlate the nitrogen demand with the fruit count as established on the image on the right, which was received by an app called Outfield, and we also did them using aerial imaging. So all right, we have our demand, but what about supply? We used soil optics in order to actually find the organic matter or the concentration of organic matter in soil. And luckily, that also correlates and it's linked to available nitrogen. That's why we can find a rate of nitrogen. So for the future and in this project, our goal is to combine these 2 values and actually find a prescription map using Outfield. And that's it. For my acknowledgements, I would like to thank everyone at the Salado Lab, at Agade, and our cooperators for helping make this project possible. Thank you.